and you can then see it. You guys can hear me?
Yes,
Yes.
we can hear you.
Okay,
Yes, I can hear you.
Okay, awesome. I just actually got a notification that my signal was unstable. So I just want to make sure that you guys can hear me. Okay, so, you can see all kinds of detail. You can see, algal colonies. And in terms of the detail that you're seeing with each one, is going to depend on the quality of the lenses as well as the limit of resolution, right? So there are two main concepts when you talk about the light microscopy, right? One is magnification and the other is limit of resolution, right? And so, can somebody tell me what is the concept of magnification and how can we figure out magnification for any given objective lens?
So the magnification of objective lens is 10%, and then like the arms in the spinning thing also have magnification, and so we multiply together to get the total magnification.
Okay, all right, so you mean the ocular lenses, right? So those have the 10x, they're generally fixed. That's the only one you have. So you're right, that's the only one. Generally it's 10x, but you can have other microscopes that can have like 15x, or it can change, but generally the ones that we use in laboratory setting absolutely have 10x ocular power. So they individually can magnify 10 times, and then you can have multiple objective lenses on the sort of the nose piece, and then depending upon the objective that you have in place, you can have multiply the power of the objective with that of the ocular to get total magnifications. Thank you so much for that. So yes, that's how you calculate total magnification. So you should be able to manipulate that, right? I mean, if somebody gives you total magnification, you should be able to, and the ocular magnification. If it's not given, you always assume it's 10x, right? So you can calculate the magnification of the objective lens very, very easily. So these objective lenses have a certain quality to them as well, and that quality is referred to as a numerical aperture. And numerical aperture is basically the ability of a lens to collect the light rays so that they don't bounce off, okay? And so the better the lens, the better the numerical aperture, okay? And they each come with their own value, okay? And you can certainly look them up. Or I can give you, for example, a standard set of numerical aperture, and I can ask you to calculate the limit of resolution. Does anyone know how the limit of resolution is calculated? Anyone? No one wants to volunteer that information? What is limit of resolution? Just resolution, resolving power. Anyone knows? Andy, you want to try? No? Kiana, you want to try?
Are you asking?
Cameron, you want to try? Oh, Julia's going to try. Okay. Who was it? Sorry.
Oh no, I was gonna ask if you can repeat.
Go ahead. Don't be shy. It's fine. I'm not. So what's the maximum? You'll get it wrong. It's not a big deal. Let's talk about it. What is it?
I'm sorry, are you asking what resolution is?
Yes, let's talk about resolution and then how to calculate
general-
resolving power of a given microscope.
In general, I think it's just like, in general terms, the ability to distinguish like fine details.
right? So how clearly you can see an image. So for example, with this particular one, looks like a diatom or some algal structures, and you could see these little beads on their surface. So that's a very fine resolution. You can really see the details on it well, right? So that image was, if it's an actual image, it's actually a taken with a microscope that has a very high power of resolution. And so generally your electron microscopes would give you a very high resolution like that. Okay, you can see larger structures still with some fine detail, right? There are several colonies, smaller colonies that we can see within this larger colony, but we can't see the finest detail there is because, that's beyond what this microscope could have resolved. So power of resolution is just how clearly you can see detail. Yes, Olivia.
For the pictures that you posted here, which one or do any of them have an oil immersion magnification?
I would think that the red blood cells are probably under the oil immersion,
Okay, thanks.
but it doesn't, you can't tell just by looking at the picture which one it would be. The oil doesn't do anything to the image other than just allow light to pass through a little bit better. Okay, so if you wanted to calculate the limit of resolution for a given objective lens in a microscope, does anyone remember how that's done? What kind of information you need?
I'm not 100% sure, but is it the formula of wavelength divided by 2 times numerical aperture?
Yes, it's exactly that. Okay, so generally, the microscope that we uses white light, right, which has a range of wavelengths. So if you wanted to calculate the resolution, then you would have to calculate it for individual wavelengths. So you could have the blue light wavelength, which is going to be a lot shorter than, say, the red light wavelength, right? So you would have to do monochromatic light. So the light source should just give either, say, blue light or red light or orange light or green light, whatever lambda you choose, which is wavelength. And then you need to know the numerical aperture. Okay. So let's say, okay, you have an objective lens that has the numerical aperture of 0.4. Okay. And you're using red light. Okay. Can you guys figure out what the resolving power would be using that formula? Okay, so the formula that was given was resolving power equals wavelength of light, which is lambda, over 2 times NA. So there are 2 variables here, right? One is the lambda, which is the wavelength of light, and the other is numerical aperture. Okay, so I'm telling you, let's say we're using one objective that has the numerical aperture of 0.4, okay? And then we're either using red light or blue light to look at the same specimen. Everything else is the same. The difference is the wavelength of light, okay? Can you guys calculate the resolving power for the 2 wavelengths? Let's just choose standard wavelengths. Everybody has the same values. Let me put it in the chat. Okay, so let's see, red is around 750, okay, and let's see.
Professor,
Yes.
I just want to make sure you're not drawing anything, right? Or showing anything on the slides.
What do you mean? I'm not drawing anything or showing anything, no.
Okay.
I'm putting it in the chat right now, okay?
Okay.
So let's say red is, the wavelength is 750 nanometers, and blue, let's say just 400 nanometers. Okay, so that's the wavelength. And the numerical aperture for this objective is, let's say, 0.4, okay? And remember the formula for limit of resolution or resolving power, you can call it either way, is lambda, which I can't draw, so lambda over 2 times NA. Okay, so let's figure out the limit of resolution for those two wavelengths. How are we doing with that? Is that complicated or we're okay with that? If it's possible for me to show work on screen. Okay, let's see. Okay, so the formula was okay, so limit of resolution equals wavelength of light, which we represent as lambda, divide that by 2 times the numerical aperture. Okay, so for the red wavelength, which we said was, let's say 750 nanometers, we divide that by 2 times 0.4. What do we get?
I got 937.5.
Okay, that's what I get. Okay, 937.5 nanometers. And then you do the same for blue and you get 500.
Yeah, that's what I got too.
That's the values we get. And what do they mean? Does anyone know what they mean now?
I'm not 100% sure, but is it the higher the value, the more clearly the detail would be? Or no.
No, the smaller the value, the more clear the detail would be. Okay, so you can have so what it's basically saying to you is that if you were using blue light, 2 points would have to be 500 nanometers apart. That's the smallest distance for them to be apart so that you can see them as 2 individual points. Okay, if you have a poor microscope, it's going to just show you an oval. All right, so if I were as if these 2 objects were merged together. Okay, these 2 points were merged together. If you were to use blue light, okay, what it's saying to you is that they can be a certain distance apart, okay, so that you can distinguish them as 2 separate entities. Okay, so that's, let's say, 500 nanometer distance. Now we can see, okay, that with blue light, what we're saying is, I'm sorry, with the red light, we're saying is that the limit of resolution is 937.5 nanometers. So what we're saying is that the objects would have to be about twice the distance apart under blue light for you to be able to see them as individual entities. Okay, so they would have to be farther apart. So you can use the same lens, right? But you can use two different wavelengths of light to illuminate your specimen, and you can see, you know, detail better. Okay, so the smaller the wavelength, the better the detail. Okay, and the smaller the number that you're getting, smaller the power of resolution, smaller the distance between them, and the better you can see them. Okay, and so if we continue to then think about ways of illuminating our specimens and then ways of making that wavelength of light that illuminates it smaller and smaller, and there are various ways of doing it, right? You can have laser light, you can have, you know, a beam of electrons that's going to have the shortest wavelength, and therefore we see the greatest detail with the electron microscope. Okay, so power of resolution is a function of two things: the wavelength of light that we're using to illuminate the specimen, and two, the numerical aperture, which refers to the quality of the lens, of the objective lens, and how well it captures light that strikes it. Okay, the more it captures, the higher the numerical aperture. Okay, the more light is reflected and lesser is allowed to enter, the lower the numerical aperture. Okay, so you can't control numerical aperture. You can just swing in a different objective lens with a better numerical aperture, right? So generally when we look at, and I want you to look this up, you know, as we go through our objective lens, the numerical aperture gets better and better and better, okay? And the oil immersion lens combined with oil would have a numerical aperture of 1.25, okay? And so it gives you the highest quality image, the greatest resolution, because combined with oil, that lens is able to capture a lot more light, okay? And so the resolution is going to be a lot better. So it's not just an image that is magnified and looks all blurry. It's an image that shows you fine detail, okay? Questions on that? Okay, so you should try, and maybe I can make it a part of an assignment, try maybe using two different, maybe same light, let's use blue light, and let's use two different objectives, one with numerical aperture of 0.4 and the other with the numerical aperture of 0.6. And let's see how that changes the power of resolution for that microscope. Okay, so let me see if I can change that activity for you. So give me 1 minute. Okay, so I just updated that. So there's a question related to limit of resolution. Any questions you have so far on this particular concept? We're good? Say something so I know you guys are here and you can hear me, because otherwise I just kind of feel like, am I not connected in chat or somewhere?
Yes, we can hear you.
Okay, awesome, thank you. All right, so let's move on. Okay, so you want to know the limit of resolution. Okay, something funny. Okay, I don't know if you can see all of those angry faces of Xylem and Phloem. Yeah, no? Okay, not funny. All right, that's fine too. All right, so who can tell me what is the proper method of, like, the sequence of events of using this bright field light microscope? Like, what do you do first? And then how do you go about it? Now, let's start with, so it's plugged in already and the light's been switched on, and so you want to go in with the slide now. What do you do? Yes,
Um,
April. Oh, awesome. There's multiple responses. Cool. Who wants to go first?
You can go first.
Okay. All right, guys. Who was it?
That was me. Don't you have to make sure it's on the lowest magnification scope?
Okay, cool. So that's the first thing, right? So that you want to make sure that the lowest magnification objective lens is in place. And then before that, you need to put the slide onto the opening on the stage, right? So that, and you control the light and so on. So that's certainly also part of that. And then, yes, you want to adjust the ocular lens for what?
To make sure that it's one image.
Right, so that it's going to adjust the distance between your eyes, because everybody has a difference in terms of how the two eyes are, how wide apart they are. So you can control the distance between the lenses, and so that you can see a single image, single circle, single field of view rather than multiple ones, right? And then you can even have, you know, these adjustment rings on the ocular itself that help to make the image a little bit better. So you have the ocular lens. You don't have to, should you be wearing glasses if you wear glasses generally? Or would the microscope fix for that?
I believe you don't have to wear glasses.
No, right. You don't wear glasses because it doesn't matter whether you're nearsighted or farsighted. The mechanism would adjust for that, you know, difference in accuracy. So that would be fine. All right. And so now we have the objective taken care of. We have the ocular taken care of, and now you wanna begin to focus on the image, right? What do you do?
You make sure the illuminator is on at a good brightness, and then you turn your
Okay.
coarse focus.
Okay, so which one do you start with? Course, right? So you start with
Course.
course. And yes, absolutely. So you are gonna, generally the way to do it is that you would, if it's the stage that rises, or sometimes, you know, it depends on what moves. Generally, it's the adjustable stage. So stage moves. So generally, you would move the stage as far close to the scanning objective lens as you possibly can, as you're looking at it from the side, so as to not hit the slide and crack the slide and break the slide, as well as damage the objective lens. So you want to be looking at it. And then you begin to lower the slide as you look through the oculars, so that you begin to, you know, slow down as you are seeing a sharper and sharper image. Okay, so you want to raise up the stage as close to the objective lens as possible while you're looking at it, but then you should be looking through the oculars when you begin to lower it. Okay, because lowering is not gonna associate with crashing the slide and so on. So that's the first step. And once you see the defined image, then you can begin to switch your objective lenses. And once you've switched over to 10x, which is the low power, and 100 or 40x, which is the high power or the oil immersion lens, right? Which adjustment knob should you be using?
The
You would use
fine
the fine
focus.
adjustment.
Yes, absolutely. Only the fine adjustment. And what's the reason for that?
If you're in the 100x oil immersion, you could possibly like risk cracking the lens itself or even the glass the specimen is under if you're not using the fine adjustment alone. Because using
Absolutely.
the coarse would like move the stage way too much.
Yeah, it moves it very abruptly and quickly, right? So you want the fine adjustment, you're making very, very small incremental movements of the stage. And so, you know, you don't risk hitting the stage with the objective and, you know, risk breaking it. So, you know, once you have oil immersion, before you swing in the oil immersion lens, you want to put a drop of oil directly on top of the specimen, right? When we work with bacteria, we don't apply a cover slip. If you're working with eukaryotic microbes and, you know, you're working with fresh specimen, we do. But when you're working with fixed bacterial specimen, we don't add a slide to it. We just add directly a drop of oil to it, and then we swing in the oil immersion lens. And there has to be a continuous film between the lens and the slide. If you don't have that, then you have several surfaces that the light has to pass through, right? It will exit the stage, it will enter the slide, it would enter oil, it would enter air, then it would enter the objective. So the idea behind the oil immersion lens is to have a continuous film of oil between the light and, between the slide and the objective lens, so that the light passes through the oil immersion lens always. Okay, we're going to talk about why that's the case. And one of the things that you want to always remember is that the only lens that should get oil is the oil immersion lens. None of the other lenses should come close to oil. And so you want to make sure that once you have oil on the side, you never swing in for 10, 40x. At maximum, you can do 4, because there will be greater working distance between the slide as well as the objective. But as you swing in 10 and especially 40, the working distance reduces, and so you're gonna like saturate the 40x objective with oil, and it's not designed to work with oil. Okay, and so if you don't clean it, you know, you forget about it, you're never going to see any image under that objective for the remainder of the class, okay, or for the remainder of the semester. Okay, so unless you go back and you wipe everything down, which using these special wiping paper and sometimes ethanol. And so you, with the oil immersion, then once you've applied oil onto a slide, you really can't use it under 10x and 40x. So we're going to have to then use it directly under oil, which can get very tricky to find the image, right? The whole purpose of starting under 4x and the reason for us to call it scanning objective lenses, so that you can see a larger field of view. You can scan for what you need to further zoom in on, right? And so, you know, make sure that that's the series, the steps and that you follow to be able to then eventually find the image under 100x as well. Okay. And of course, the parts, I'm sure you guys are aware of, because like you guys just used all of those terms. So, you know, are there any questions on any of this that you want me to go through? Okay, so I guess the only thing that I would just quickly go through regardless is the diaphragm, right? That is a mechanism built under the stage that controls how much light enters the stage. Right, so you're going to manually, there's a little lever there that you manually control to increase and decrease the light. So you guys have used microscope in lab. Is there like you know, something you notice about the amount of light as you go from lower to higher magnifications? Does it increase? Does it decrease? Does it stay the same?
I think it gets brighter and brighter as you increase the magnification. So you have to keep turning down the light as you go.
So it gets darker and darker, you mean then, if you need to turn up the light, right?
Honestly, I can't remember. It's been like since 2019, but I believe so.
Yeah, yeah, that's fine. All right, so if you need to turn up the light, that means that the image got darker. So you went from 4 to 10 and 10 to say 100, you know, sequentially, of course, the image would get darker and darker because you're zooming in on a very small portion of it, and you might not be collecting as many light rays coming from that area. So you have to continue to increase the intensity of light. Okay, so diaphragm is ideal for that purpose. All right, and so stage setup. Yes, April.
I was just going to ask you about the stage adjustment knob.
Okay, yes, so stage adjustment knob basically allows you to, I believe, so this one, I'm trying to figure out if this is the one that helps you move the slide around, or is it the one that, I think that's the one. So stage adjustment knob is going to help you move the slide without having to manually push it around. Okay, so it's a mechanical stage. Notice how the slide is held between these clamps, and if you clamp it well enough, then you should be able to use the stage adjustment knob when you're trying to scan the slide, move it around to see where the object that you might be interested in is. And so it's a mechanical way of just moving around the knob. I'm sorry, slide. Okay.
Okay, thank you.
Yep. And in terms of the microscope care, there's a particular way you hold it. You always put your hand under the microscope. You hold it from the arm. If there is a wire, you wrap it around the base. You don't allow it to dangle. Of course, you turn on the light bulb always before you unplug it. You raise the objective lens as far away from the stage as possible. You always have the lowest magnification in place. You should push in the mechanical stage knob. So there are, notice here's a different kind. So this is what I'm used to seeing, and that's why I was kind of thrown off a little bit. This looks a little bit like what some microscopes have to remove the diaphragm mechanism and clean it. And so I thought it looked like that. But the ones that we would have in person, if we were to do this lab, were similar to this. So the knobs are a little bit different and located in different places. But so here, if the stage is that mechanism is protruding out, you want to make sure that you move it back in and you center it so that it doesn't break when you return it to its cabinet. So, and in terms of cleaning, there is special paper that prevents the scraping of the ocular lenses. So it's specifically designed just for cleaning these objectives and so on. So we use that. And sometimes we use things like acetone and alcohol to clean it, but that's not recommended a lot because they can also damage these lenses and the quality of the lenses. Here's the light of the path of light, originates from a light source, passes through the condenser, passes through the stage, through the specimen, and then through the objective and the mechanism and the nose piece, and then eventually out the ocular lens, and that's where you see it. Okay, and so in terms of these, working distance essentially is the distance between the objective lens and the slide itself, and as you swing in higher and higher objective lenses, you know, they are going to reduce the working distance. So you're going to have to be very careful even when you're using the fine focus knob, because it can actually still crack the slide rather, as even if it's moving it very incrementally, it can still damage it. Field of view is the area that you see under a microscope, and the field of view, of course, decreases as you increase magnification, right? You're zooming in more and more and more on a smaller and smaller areas. Your field of view is greatest at 4x and smallest at 100x. And depth of focus is sort of the three-dimensional structure of the specimen. Okay, so when you look at it under the lower magnification, right, you can zoom in on it, the entire object, but you can't zoom in on various thicknesses within that specimen. So if you're looking at, say, 2 layers of cells, you can see those 2 layers individually by zooming in on one depth where one layer is, and the second depth, which is the layer below the first layer, right? So you have this depth of focus that changes as you change magnification. Okay, so it depends on the thickness of the sample that you're looking at. And so the depth of focus reduces as you increase magnification, which means that you can optically slice this image and you can look at it in various depths. So if you've looked at ever the cross thread phenomena, where you look at the red thread and the blue thread and the yellow thread, under 4X, you can't really tell, which one is on top, or, you can't focus on just yellow if you wanted to. You would have to focus on all of them at once. But under 100X magnification or 40X magnification, you can focus on yellow alone and the other two become blurry, and you can focus on blue alone and the other two become blurry. So it's basically telling you that you can focus in on the various depths within that specimen. Okay, and the contrast is just going to actually, in terms of bacteria, we're going to add stains to increase contrast. Generally, if you have a brightly lit background and you have microbes that don't really have any color to them, they're colorless, they're translucent, the light passes through them and it bleaches out the specimen, so you don't really see anything. So in microbiolab, staining is a big part of the technique. So we're going to talk about it next, I believe on Monday or Tuesday, rather Tuesday or Wednesday. But it's going to give it that contrast. So we're going to use dyes that will adhere to the cells, make them colorful so that the object stands out against a bright background. And I've already talked to you about the resolving power. So this is just kind of, again, showing you what's going on in terms of field of view. You go from greatest, which is at 4X, to the smallest view under 100X, as if when you're zooming in on something. And power resolution increases with the numerical aperture, and the higher the magnification, the better the objective and better the numerical aperture. And focusing knob, notice you only use coarse focusing knob under 4X and only use fine for the remaining. And depth of focus, as I said, you're going to have the reduction in depth of focus as you increase magnification. Okay? Questions on any of this so far?
Professor?
Yes.
I believe with the 10X, I was taught to use the course for the, for the 10X, use the course instead. I don't know.
No.
All right.
Just under 4X, unless you did not have 4X.
Okay.
It's possible that sometimes microscopes do not have the 4X objective lens. And so if they don't have it, then you would be forced to use course under 10X.
Okay, thanks.
Yeah. Yep. Okay, so oil immersion lens, what is the concept of oil then? If we were looking at these microbes under the microscope in lab, there's no way we're going to see much under 10X or 40X. We really have to look at them under 100X so that the total magnification is 1,000 times, right? So you're looking at something 1,000 times bigger at that magnification. And generally, when light is passing through a specimen, so here you have the slide and here's a cover slip. In our case, we're not going to have a cover slip, so we pretend that that's not there, but either way, it doesn't matter. With an objective lens that doesn't have oil, what we see is that as light passes through various media, like it passes from glass to air, and then again back to glass, and from glass to back to air, and then eventually enters the objective. So that's the path it has to pass through. And the way light works is that every time it changes a medium through which it should travel, it will disperse. Okay, some of the light waves will bend, they will change their path, and because they change their path, those light rays will not enter the objective lens, they will be lost, okay? And that light is extremely important, not just illuminating the specimen, but also seeing fine detail, right? So as you increase the magnification, there is smaller and smaller distance, and the objective lens have to be better and better at trapping light, okay? As well as just the quality in general so that you can see detail, okay? So the objective behind putting a drop of oil immersion lens is that you place it on top of your specimen, and the oil immersion lens, I'm sorry, the oil that is used for this purpose is actually, it has the same refractive power as the glass itself, right? So refractive power is basically the ability of a medium to bend light. So what that means is that now when light enters the glass, right, whether it's a cover slip or slide, and then it exits it and then enters the oil, because the refractive power is the same, light acts as if, you know, it's still going through oil. It treats it's the same as the glass cover slip. So none of the light rays would bend and none of the light would be lost, or a lot of the light rays would actually keep traveling at the path that they originally were as they exited the glass cover slip. So because it's straight ahead, a lot more light than would be captured with the objective lens. So you can't use a oil immersion lens and see a very fine focused image unless you put the oil immersion lens on it, right? So it will magnify it. It will just not give you the resolution that you're looking for. Okay, so you want to be able to see bacteria, and the maximum really that you can see is their shape, arrangement, and of course the stain that you have applied. We're not going to be able to see their flagella and pili and things like that unless we somehow deposit salts on them to make them look thicker and so on. So we'll talk about that when we talk about staining, but you couldn't just swing in a oil immersion lens under dry conditions where you haven't put in the oil and expect to see a fine detailed image. You're not going to see it because it works only when you put the oil onto the slide. And notice when the oil is added, it's a continuous film. You can't have a space because that will introduce another area for light to bend. And that means that the objective lens then has to be really cleaned before you leave for the day, unless you want like a thin and film to add every time, and then a thick deposit to form, and then eventually you will lose visibility that way as well. Yes, Olivia.
Sorry, just to clarify, so for 4 times magnification, it's coarse, 10 is fine, 40 is fine, and then right before 100, like right before in the middle with them sticking out side to side, you put the oil and then you switch it to 100.
Yeah, and then you swing in 100, yes.
Got it.
Yep. Okay, so any other questions?
So do you put just a very small drop of oil?
Yeah, so it's not a very, so it comes with a dropper, and when you add a drop, it will add enough so that you can have a film. You can look at it from the side and make sure that there is a continuous film between the slide and the objective. If not, then you can add more, but of course it can't be flowing around everywhere. So there's very little working distance you're working with when you have this 100x objective lens, so it's easy to get there with a big drop. Yeah.
Okay, thank you.
Okay, so when you look at it, look at an image via oil immersion lens, you can, here is an image where the oil wasn't added, and notice you magnified it. It still looks the same, but you can see a lot of detail. And here now, a drop of oil was added, and now you're beginning to see a little bit more detail. I'm seeing blood cells within a vein, and I'm seeing some, tissue and some connective tissue, and I'm seeing some nuclei that are stained purple. So I can see a lot more detail now. I can suddenly recognize what it is. But with this one, it's still blurry enough so that I can't be certain as to what I'm looking at. Are these bacteria or are they really nuclei? I mean, I'm comparing it here, so I know that they're nuclei, but if I were to see just this area, I wouldn't be able to discern what it is, okay? So you're gonna get much better resolution because many more light rays are being captured. Okay, so, all right, so hopefully you got that. If not, it's all right. All right, so fluorescence microscopy is basically, it's going to give you these images that are brightly colored, very pretty. You can use, fluorescent compounds that give off a certain wave length of light when you illuminate them with laser light. So you have to first shine some laser light onto them, and then they absorb some of the wavelengths, and others they return, right? They reflect. And so the reflection is what you're gonna see, right? The colors red and yellow and blue that we're seeing here in this image. So these are fibroblasts that have been stained for DNA with the blue fluorescent stain, and the microtubules with green and actin with red. And so there are various ways of accomplishing this, and we actually have a cell bio lab where we do this technique, immunofluorescence, because we use antibodies to deliver the fluorescent compounds. Okay, so there are various ways of doing this, and so there could be proteins that fluoresce as well. So green fluorescent protein fluoresces green, so we can fuse that to proteins and then track that using fluorescent microscope. So it looks the same and the mechanisms are the same, just the light source is going to be different. Okay, so that the light source can sometimes be just bright light, other times it could be laser, but we won't necessarily call it then the fluorescent fluorescence microscopy. Okay, so it is similar to bright field, but the image that you're seeing is coming from compounds that themselves fluoresce, and we've used those compounds to target particular proteins within the cell itself. So here 3 separate compounds were used to label 3 separate parts of the cell, and then you had taken 3 separate pictures that then computer is able to superimpose to give you a pretty picture like that. Okay. And so then you also have a confocal microscopy. Confocal is often gonna use laser light. Okay, fluorescence microscopes can also use laser lights, and they're gonna basically take a specimen and they're gonna optically section it, right? So you can have a cell that's been sectioned into thin slices optically, like you don't physically actually cut it into 10 thin slices. You can focus at various depths within that cell, and you can have 10 different levels that you can focus in on, so you can see detail that right here we're seeing some of it, but you can see in this case, for example, we're seeing part of the I believe what looks like the brain, but certainly blood vessels and the particular cells that they are serving. So, within the cell, you could see, if it was, say, a yeast cell, you could optically section it and see nucleus in a lot more detail, or mitochondria in a lot more detail, and so on. So it's gonna use laser light. It's gonna give you very high resolution images. It's gonna give you very high magnification, and the ability to look at it, at the image at various depths, and then not only see those individual sections, but also have the ability to combine those sections into a 3-dimensional image. So that's what like confocal microscopy does. And then in scanning electron microscopy, which not every lab has, it's going to use a beam of electrons to illuminate the object, and the specimen has to be prepared very, very differently. Generally, the specimens would have to be prepared using heavy metals so that they actually trap some of those electrons. And so you can have the dense looking regions and lighter regions, and dense regions would be where, the electrons are absorbed, and the lighter regions are where the beam of electron passes through. So in scanning electron microscope, we're just looking at the surface of the specimen. We're not penetrating the surface. We're not sectioning the specimen. We're not looking at the inside of the cytosol. So you could have like a head of a mosquito, for example, that you want to take a picture of in fine detail and see what its eye structures look like in finer detail. So you would use electron microscopy, because you can magnify up to 100,000 and more times using scanning electron microscopy. So much better resolution because of the low wavelength of electron beam that we're using, and very high magnification as well. And then we can have transmission electron microscopy, where we're actually looking at the intracellular detail. Okay, so you might be looking at a lot of viral pictures as we go through this slide, and what we're going to see is detail. So viruses don't have a lot of detail. This is a very fine resolution image of a virus, and we're seeing spikes around it, and we're seeing the protein shell, and then there's something within that dense something that that you could see is actually its genetic material. So it's going to give you sort of a two-dimensional image, and it's again going to be black and white, just like you would see with the scanning electron micrograph, but then we can add images using computerized, we can add colors to the images using computerized tools. Okay, questions on microscopy? No? Okay, so what I want to do is, there are a couple of things we're doing today. One thing that we're doing today is the calibration of the ocular micrometer. And so we're going to use a virtual microscope for that. The other thing that we're doing is a couple of Connect simulations. So why don't you guys go ahead and do the Connect simulations. We'll be back here in, say, 20 minutes or half an hour. Let's say half an hour. Let's do your simulations. Watch the video if you have to for how to function a microscope, and then we'll come back and then we'll do calibration. Okay? Sounds good?
Yes, sound good.
Okay,
So
so
like-
let's. Yeah, go ahead.
Do we stay in the room, or should we
still
come
stay in
back?
the room. No. You stay in the room, but we'll kind of start coming back here and talking about the calibration. But please stay in the room. You are free to talk to each other, talk to me. I might go in and out, but I'm here. I'm not leaving the room.
Professor, you said lab simulations on Connect, but I
Yeah.
can't see any other simulation besides like all the sessions I finished.
Really? That's-
Yeah. The last one is a broth to slant.
So you don't see the bright field microscopy and oil immersion lens? Did you already do them?
On a Connect?
Yeah.
Yeah, I did that.
Okay, so then you're just gonna have to wait a little bit
okay.
until everybody else is done, and then we'll talk about the ocular micrometer. So you can start looking at that activity. Just click
Okay.
on the website and start reading through it in the meanwhile.
Okay, thank you.
Yep.
Professor,
Yes.
I think the slide you post on Google Classroom is not the same
that you
Really?
be saying. Yeah, because I saw there is multiple style. We don't have it.
Oh, well, it might be an older version because we used to do different things in person. So I'm gonna change that. I apologize.
Okay, thank you.
Please try again and see if you now have the right ones.
Hey, Professor, I just have a question for the test your knowledge for the lab. So for the paramecium, are we gonna go over that a little bit?
Yeah, so that's the one that we still have to do. So in the
Okay.
meanwhile, just kind of read the calibration, go to the calibration page and then start reading up on that. And then we will come back here together and talk about how to do it.
Okay,
Okay.
perfect.
Yeah, I think it's the wrong PowerPoint again. I don't know why is it the wrong one. I'm gonna try again. Okay, so I updated it now. I think it's the correct one. Just please confirm that it is. If it's not, just let me know.
Professor,
Yes.
is this activity only posted on the Google Classrooms?
Oh, yes, it is. So hold on. You mean the calibration one, right?
Mm-hmm.
Yeah, yeah. I'm gonna post it on Blackboard right now. Thank you.
Okay, thank you.
Professor, I have a question.
Yes.
I see that you posted the 2 short videos about the agar plates. So are we supposed to melt the agar into some pot?
So, yeah, so you're gonna, so there's a rack that came with the kit, so you can put your agar test tubes within that rag and then place it into a pot, and it's not going to get contaminated because this agar is actually sterile. Okay, so you can rinse the pot afterwards and there's no issue with that.
And did the kit come with like the Petri dishes?
Yeah.
Okay, perfect. I was just wondering like where we should put the melted agar, but if it comes with it, it's good.
Yeah, absolutely, absolutely. Just make sure when you take out the test tube from the water that it's not dripping, and then you don't end up pouring that dripping water into your agar, because it will get contaminated then and you will have growth there when you shouldn't. Just be very careful with anything that is outside of what's inside the test tube, right? Getting
Mhm.
in plates.
I see that it says it takes 15 minutes to melt the agar, and then it says it should take 30 minutes or so. So which one, how long should we do
Oh,
it
I
for?
think as soon as you see it get translucent, I think you would get an idea of that it's melted already. If you don't see any lumps, anything floating in it, any
Mhm.
lumps of the agar remain. As long as you just see a fully fluid translucent agar, that's it, you're done. So
Okay.
it could take less than 15. I don't know. It just depends on how hot your water is, and you don't just want the agar boiling, so it's going to take some time to melt, and it shouldn't be boiling during that time because you'll be losing a lot of the moisture in it, and you don't want to
Mhm.
lose. So do it to bring the water to a boil, and then just let it sort of not even maybe simmer just very gently. Turn down the heat at like low medium so that it's not vigorously boiling, but it just stays warm, and then just leave the stand in there. And try to put the stand, I would suggest, away from the center, away from the direct heat. And don't put it in there as you boil it. I would really recommend first bringing water to a boil, and then just introducing the only issue with that would be that I hope that it doesn't crack, and that I I have to keep in mind that you guys are not necessarily adjusted to that. So I guess just leave it from the very beginning so that it slowly is brought to boil. Don't put in like high flame under it, just slow, medium, and bring it to boil gently.
Okay, sounds good.
I'm still transferring it to Blackboard, so give me a minute. Okay, posted on Blackboard now.
Thank you.
Professor, I'm sorry, I have one more question.
Yes.
So I see in the video for the agar plate, she used one tube for 2 Petri dishes. So how many tubes should we have for like a slant, like culture swab?
So what we're gonna be using it for the first lab, right? And for that you need, I believe, 3 petri dishes. Let me just confirm that, okay? Because I read it yesterday and I just want to make sure that I'm giving you the most accurate information. Give me a few minutes.
Okay.
Hello, Professor.
Yes.
There is there a post lecture assignment for today?
Yes, there is. I posted it just now.
I can't really see it. Give me a second.
Maybe reload your page.
Yeah, I'm only seeing the session 1.
Really? Okay, give me a minute. Do
Johnny,
you mean for the Blackboard?
Yeah, for Blackboard.
Yeah, Blackboard, I'm gonna post it right now. That's what I'm in the process of doing.
Oh, okay, okay.
Hey, Professor.
Yes.
After we finish the Connect simulations, are we supposed to do something else?
Yeah, so start reading up on the calibration one, and we'll be talking about it in another 5 minutes.
Oh, where do I find the calibration? I'm sorry.
It is on Google Classroom. Oh, so you were also looking for the Blackboard one? I just posted it.
Okay, thank you. Hey, Professor, I have another question.
Yes.
I heard some people were talking about the petri dishes. Like, are we supposed to start working on that too?
No, so don't work on that right now. That you should do after class.
Okay.
Unless you want to do it with me and with me here, just so that you're sure that everything goes fine, that's fine too. But let's first finish today's lab. So let's do the calibrations. Give me a few minutes. I'm just gonna quickly look up how many we're supposed to pour and how many tubes to use. So I will let you know in a minute.
I appreciate it. Thank you.
Okay, so regarding the petri dish, we're gonna melt a single tube and we're gonna split the contents into 2 petri dishes. Okay, so half and half. So if you want to mark it somehow so that you know how much is half. Okay. Are we here? We're good? Did we get it?
So just 1 tube for 2 Petri dishes.
Yes, so you're gonna melt the contents of 1 tube and then just either eyeball it or actually physically mark where the half would be so that you don't have pour too much into one and too little into the other. You want to pour equal amounts into the 2. They're tiny, so it should still give you a pretty good thickness.
Okay, perfect. Thank you.
All right, so how many people still need time to do the Connect simulations? Because otherwise we can talk about the calibration because that can take some time too. Anyone who needs time still for the calibration? I'm sorry, the simulations?
I'm ready to talk about the calibration.
Okay, thank you. April, anyone else who needs time or we're good to go? If I don't hear back, then I assume we're good to go. Okay, cool. All right, let's go. All right, so let me just pull it up. Okay, folks, so the calibration of the ocular micrometer, what we're basically doing is we're going to learn the distance between the markings on a ocular ruler in standard units. So when we actually use those to measure how big something is, then we can call it, you know, a micrometer or a millimeter or a centimeter, right? Right now, if you've used microscope in the lab already, what you generally see is a ruler in one of the ocular pieces that reads from 0 to 100. Okay, do you guys remember seeing that at all?
I remember seeing it.
Yeah, absolutely. There's every microscope that is standard laboratory microscope generally has one eyepiece within which you can find a ruler, and then you can use it to measure how large something is, but you can't give it any standard units because it doesn't contain any units until you calibrate it so that you know the distance that in standard units between the these two adjacent lines. So that's what we're going to do. We're going to use a stage micrometer, which actually has standard units, okay? And it's generally, you know, the entire thing is 1 millimeter, okay? And it depends on the micrometer that we might have. And so it's generally 0 to 100, where 100 denotes a single millimeter, right? So you have, you know, 1 millimeter long micrometer that's going to be on a slide. And then we're going to use that to match up against the ocular ruler lines and then figure out the distance between the ocular ruler lines so that we can figure out then, you know, the calibration and then basically use it to then begin to measure. So when we look at ocular micrometer, no matter the objective lens that you have in place, it doesn't change size. It remains the same, okay? And the one that you have on this stage or on a slide, it's gonna continue to get larger and larger, right? Because you're zooming in on it. So under 4x, you're gonna see most of it. Under 10x, you're going to see a portion of it. Under 40x, you're going to see even less. And then under 100x, even, you know, less. All right. So because you're zooming in on a small portion, but notice how, you know, here you're seeing the stage and the ocular ruler. Here you're seeing the stage and the ocular ruler, but, you know, the two are not the same. Here under 10x, the stage ruler looks smaller, but under 40x, it looks a lot larger. Okay, so what we're going to do is we're going to match up one end of both the ocular ruler and stage micrometer. And we're going to use the stage micrometer as an actual ruler to figure out how many of these lines can fit in here. So if we look at this, we have 0.1 millimeter, 0.11 millimeter, and 0.12 millimeter. Okay, so you have 0.12 millimeters that equal 51 lines on the ocular micrometer. Okay, so then you can easily figure out if 0.12 lines or 0.12 millimeters equal 51 lines, then how many millimeters would equal 1 line, right? It's a simple proportion question. And so you can easily figure that out. So we're going to use I'm gonna, sorry, go back. I'm gonna stop sharing and I'm gonna share the screen now where we're using the calibration. So everyone should be on this website, okay, microscopy website. And we're basically, oh, what happened? Okay, give me a minute. Oh, wow. I'm having trouble finding it. Give me a minute. I don't know what happened to the website. It almost seems like it's not there anymore. Give me a minute.
When I went to the website, I was able to access the calibration, but then I think it's your browser.
Yeah, exactly. I just changed my browser, so don't use Google Chrome. Try another browser. What am I using right now? I'm using Microsoft Edge right now, I believe. So try a different browser. Try Microsoft Edge. It's working on Microsoft Edge.
I'm using Firefox and it's working okay.
Okay, so Firefox works too. So it must be something with Chrome that it's not working. So open it using another browser. So I'm going to share it now. Okay, so you guys are seeing it on your screen, hopefully. So just go there and then we'll go from there. Okay, so everybody has it?
Yes.
I
Okay, so
have
what?
it.
All right, awesome, awesome. Anyone who doesn't have it? Okay, all right, so what we're gonna do is we're gonna pretend like this is our eyepiece ruler. Okay, notice it has these markings from 0 to 8. Okay, and they don't have any units. They're not in any standard measurement. Okay, so they're just arbitrary markings that we can use to measure. So I can, for example, move it around and say, okay, I can move my either I can move the specimen. I can't move up, of course, the ruler because it's part of the ocular lens. And so I can move that around a little bit and I can figure out maybe the width of these cells, right? So maybe this one. Okay, so what I noticed is that it's almost 3.5 units. Okay, that's what it's saying. That's all it is, 3.5 units. It's not 3.5 millimeters, micrometers, nothing. And units are not a very repeatable measure, right? These units could mean anything. So if you were reporting the width of the cell, you would have to know exactly if it was micrometers or millimeters or whatever. So what we're going to do first is we're going to figure out how many units of this ruler we can fit into a standard ruler, right? So this is an arbitrary ruler, and we're going to use an actual micrometer. We're going to use an actual ruler. And the magnification we're going to use is the lowest possible. So let's start with 10. All right. So your stage ruler is at, the red color, black color, whereas the ocular ruler is red, right? So I want you to match up on one end the two rulers and then write down in your, assignment is that? The ocular micrometer assignment, if you open that as two, there is a table there for you to fill, where you're going to write down the ocular ruler divisions, which there are 80, right? And you're going to write down how many stage micrometer divisions equal those 80. Or you can choose a smaller number. You don't have to always choose 80. You could say, well, okay, what if I chose just 10? Okay, but it's sometimes easier to just work with the whole thing. So let's first do that. Let's first figure out by matching them up on one end. Let's start with 0 end. Match up the entire ocular ruler, which is red, with the stage micrometer divisions. Okay, let's do that first. So for all objectives, 10, 40, and 100. So not all, I guess, just those three, 10, 40, and 100.
The stage micrometer is in red color, right?
Yeah, I
No,
think
it's
this.
I think the stage is in black, isn't it? The ocular was red. Let me just look again. So if you took away the micrometer and you instead put the specimen, right? So you select the object that you don't want to select micrometer, you select the specimen, then you see red markings. And so that's the ocular ruler. Okay, and when you throw in the micrometer, then you start seeing blue markings or black rather markings as well. So that black ruler is the stage ruler. And you also know because that's the one that moves when you move the slide around using X and Y translation.
Sorry, Professor, are you sharing the screen right now?
I'm
Because I don't see
sorry.
anything.
Okay, hold on. There's so many tabs open. Okay, there we go. Okay, so notice here you can select an object, either a micrometer or a specimen, right? And so right now I've chosen a micrometer, but if I select a specimen, so I'm seeing some algae in the background, now I see the ocular ruler, and that ruler is in red, okay? And now if I select the stage micrometer, now I'm starting seeing, that ruler, and I know it's staged because when I move it around, it's the black ruler that moves, okay, not the red. Red is not going to move because it's fixed in your ocular piece, okay? But this one moves, the black one moves up and down or, side to side, right? So we know which one is which, okay?
You said, sorry.
Go ahead.
Sorry, my rulers are not lining up, even though I'm
Yeah,
adjusting.
mine don't line up either. So it's not really possible to get an accurate reading.
Correct. Yeah.
So they never line up?
No.
Yeah, same here.
Okay, so can you do an approximation? No, there's no approximation either. Can you do just 10 markings or 20 markings? Try 20 markings on the stage. You just have to have, you know, something equal one of these stage rulers. And so as long as you know how many markings of the ocular ruler fit into the stage markings, then you can do a comparable proportion, and we just need to figure it out for one. It doesn't matter whether we measure the whole ocular ruler or we just measure a portion of it.
The Y translation is the issue right now for me. It's like I can't
Okay,
get
if
it.
I leave it here where I have on my screen right now, right? The 8 total markings on my red ruler equal 40 markings on my stage micrometer. Yeah?
Yes, so then does that mean that in this question 1 for the table, you want us to put for 10x ocular ruler divisions is 8 and then stage would be 40?
Right.
Okay.
Professor,
So
can
it's, I wouldn't, I guess, I mean, so there isn't 8 divisions, I guess. It's so there is, you're going to keep in mind that there are smaller divisions as well. And what we want to really measure is the distance between the smaller divisions. So let's, so you have multiples of 4 really, right? So you have a total of 28 divisions on the red, which is the ocular. So these are 28 divisions that equal 40 divisions on the stage. You understand?
Can you repeat one more time?
Yeah, so divisions are, you know, the individual lines, right? So there aren't 8 divisions. I'm going to take away the stage micrometer. There aren't just 8 division. A division is this small line that you're seeing. So there are various lengths because they give you a whole unit and then half a unit and a quarter a unit. So we are still measuring the distance between any 2 adjacent lines. So we're going to count all of them. And because there are 8 whole units and each one is split into 4, so I'm thinking there are 28 total units. Somebody can, I guess, count them if they want to, or 32 rather, not 28, sorry, 32. 8 times 4 would be 32. So there should be 32 units that equal 40 on the stage. Okay.
so for the ocular ruler divisions, are we rating 32 or 8?
32, because there are 32 total lines, and so there are 32 total divisions, 8 units, but divisions in terms of those lines that we're seeing, there are a total of 32.
I'm having trouble.
You cut off. I can't hear you.
Sorry, with the 100 objective lens.
Because it's going to get much smaller, right? The stage lens is going to get much smaller. I don't see a problem. Why do you see a problem?
It's not lining up whenever like
If you look at my screen, it's perfectly aligned.
Is that 5 units?
1, 2, 3, 4.
Thank you.
Professor.
Yes.
So then for every one of the ocular ruler divisions, it will be 32?
Right, because if you're counting the whole thing, then it's going to be 32. If you're counting half of them, it's going to be 16. So since we counted 32, then yes, it should be 32. And if that's what you're going to continue to count, then yes, for all of them, it's.
Okay, thank you.
Professor, so for the stage micrometer is 40, right? 10 times 4 is 40 division. Is that what you mean?
Yes.
Okay. How about the each ocular division in micrometer?
That's
Is
what
it
we're trying to figure out. That's what we're
Oh.
gonna figure out. So first, let's figure out what we're doing. Let everybody figure that out, and then we'll go with the rest of the calibration as well.
Dr. Chima, so from my understanding, like like each of us can have different ocular ruler divisions and we can have different stage micrometer divisions, but then for the last column, each ocular division in micrometers, it should all be the same for all of us. Am I correct?
That's correct.
All right.
Exactly.
Thank you.
Sorry, Dr. Chima, I'm not understanding that last part.
So what April said was that she could be counting, say, 10 divisions on the ocular ruler and figuring out how many of the stage ruler, divisions equal 10 on the ocular ruler, whereas you could be counting 80 and figuring out how many are there in 80, but because they're proportional, when you simplify it, the results should be the same.
So how do we figure out what goes into that last column?
You figure out what's the distance in one. You figured out how many stage units equal, the whole ocular ruler. So now you can figure out how many divisions, right, equal a certain distance. Okay, so think about it a little bit, let everybody else complete what they're doing, and then we'll talk about that.
Okay, thanks.
Is everybody done measuring? Anyone need more time? Okay, I'll give you another 10 minutes.
Professor,
Yes, Vanessa.
sorry, are we supposed to be working on part 2 of the Google Classroom activity? The one with the like the pollen grain and the diatome?
You can start working on it, but did you guys figure out did you calibrate the ruler already or?
I believe so. So I put in the ocular ruler divisions, the stage micrometer division, and then I find the ratio to get each ocular division. Is that correct?
Yeah, and then did you make the conversions in terms of micrometer yet or not?
I did, but I'm not sure if it's correct. Can I?
All right, let's do that. Let's see if anybody else still needs time to. Is everybody done with counting and figuring out the equivalent divisions between stage and ocular ruler?
Yeah.
Okay, all right. So let's then look at how to calibrate it, right? So first thing, let's do it for maybe 10x, okay? Because we saw it. So for 10x, we knew that 32 ocular divisions equaled 40, was it?
Yeah.
40 stage divisions. And we know that a stage micrometer equals 1 millimeter that has been divided into 100 units. So each unit is really 0.01 mm. Yeah. Yes.
Where is that 100 come from?
Because the stage micrometer, the length of it is 1 millimeter. The entire thing from 0 to 100 is 1 mm.
Oh, okay.
Okay, and the website may say other things, but we're gonna stick with that it's 1 mm. Okay, and that 1 mm from 0 to 100 is then split into 100 units. So then each distance between any two adjacent lines, adjacent 2 units, is then 1 divided by 100 of 0.01 mm, correct? How many micrometers in 1 millimeter? Does anyone know?
100.
Milli is 1,000 and micro is a millionth. So not 100, but
1,000.
1,000. Thank you. All right. So then you multiply that. Where are the symbols? Oh, there you are. Okay, so you get the distance between any two is then 10 micrometers. Yes, or am I? Okay, so then if the stage micrometer was 1 mm and the distance between any two lines on the stage micrometers is 10 micrometers, right? Then, and we know that 1 ocular division equals how many micrometers? Stage divisions, or I should say. So 1 ocular division would equal 40 divided by 32 equals what?
1.25.
1.25 stage divisions. Okay, and so if we know that, then now we can figure out the distance between any two ocular divisions, adjacent, of course, right? So then we know that if it's equals 1.25 stage divisions, and then we multiply that by 10 micrometers, because each stage division is 10 micrometers. Oh. Okay, you get it? So now we know that now our ruler is calibrated. Okay, so for of course that particular magnification. Okay, so we know that under 10x, if I measured something to be 10 ocular divisions, and then we did the conversion where each ocular division equaled 12.5 micrometers, then I know that if something that measured 10 ocular divisions, its actual length is 10 times 12.5 or 125 micrometers, okay? Now we only know this for 10x. Now you need to do the same thing for 40x as well as 100x, okay? Any questions on this? All right, so you guys go ahead and do it for 40 and 100x. If you look at the ocular ruler, notice it reads from 0 to 8, correct? So if it reads from 0 to 8, do you actually just have 8 lines there leading up to that? No, right? So you have between 0 and 1, you have a line that splits it in half. So there's a half mark. There's a line that gives you a quarter mark and a 3/4 mark, right? So you, for every unit, you have 4 marks, 4 divisions, correct? So you're going to count all of those. You're not just going to count the biggest lines, you're going to count all of them, small, medium, long, all of them. Just like we did for 100 for the stage ruler, okay? All right.
Professor,
Yes.
For the sage micrometer on 10x, 40x, and 100x is fixed, is not changed, right? It's also 10 micrometer.
No, so for each objective lens, it's going to differ. That's what we're trying to figure out. That's what we're trying to calibrate today. Okay, so the ocular divisions are still gonna be 32 at 40x, but the stage divisions are gonna be fewer. Okay, at 100x, they're gonna be even fewer.
Yeah, I mean the stage micrometer, the way that how you can calculate is the same on 10.
Right, right. The formula and everything else is the same, yes.
Okay, thank you.
Professor,
Yes.
so I understand that the stage micrometer divisions should decrease as you increase the objective lens magnification.
Yeah.
But because I wasn't able to align actually mine up, I was not able to find the stage micrometer divisions for 40x and 100x.
Really? Why is that?
I'm not quite sure. I can't line up the lens properly on the simulation, and I tried multiple browsers.
I'm having the same issue.
So I can't really complete the table.
Do you wanna share your screen?
I can try, but most oftentimes my computer tends to lag out when I do so.
Okay, let's try it. Yeah, you're not sharing. Malka, you wanna share?
Sure, I can try. It didn't work, right?
So you have to click on share screen and then choose the tab that you wanna share. And then hit share at the bottom.
Oh, I think I have to change something in my security preferences for that.
All right, don't worry about it. I'll share. I just wanted to show you how to maybe do it. Maybe there's something missing, but anyways, I'll share.
Sorry, it's not.
No problem. It's all right.
It's just a matter of like the fact that you know that scrolling area of X and Y, it doesn't let
Yeah.
you go further for some reason.
Yeah.
So we get stuck at like a certain area.
Yeah, same for me as well.
Oh, okay. All right. Okay. So this is basically under 10x. Okay, you see my screen?
Yes.
Okay, so you can write down what you're seeing with this, which I guess we already did. Now let's move it to 40. You don't really have to do anything. It just stays there. Just count. Should I move on to 100 for you guys?
Is it 5? Because I remember last time you showed.
Yep.
No, okay.
Professor, can I ask you something?
Say that again, please.
Professor, can I ask you something?
Yeah.
For the ocular I mean, for the style, you just teach for the ocular micrometer and stage micrometer. Why is it say that is micrometer, but the unit you said is millimeter?
Because it's a microscopic ruler. So we call it a micrometer. That's just what we call it because the microscopic units are micrometer, which is a millionth of a meter. Okay. And it because we're seeing it using a microscope, and also because the units on it are in micrometers, 10 micrometer, right? That's why it's called a micrometer ruler.
Oh, okay.
It's just the name of the ruler too.
Okay, because I keep thinking why we still need to count for the micrometer.
Yeah,
and
no.
That's already there.
Yeah, yeah. So we're really calibrating the ocular ruler, right? Because when we're going to be using this to measure a specimen, right, the stage micrometer is not going to be there. So now if I measure these cells that I'm seeing under 100x, and I notice that it's 18 ocular units, right? Now I can see my calibration value for 100x and multiply those 100 units, multiply those 16 units by that, and that would be my microscopic measurement.
Professor, can I ask you one more thing?
Of course.
For the formula you just gave up, the stage micrometer you said is 1 mm over 100. So the 100 is come from 10x times 10x, right? Oh
No. It's coming from, look, so when you look at the micrometer, I'm going to look at it under 10x. Notice it okay, it starts at 0 and it goes to 100. So there are 100 units on the stage ruler, okay? And that is where the 100 is coming from. That 1 millimeter has been divided into 100 units, okay?
Okay. Oh, now I get it.
Okay.
Professor, can we go over the question 2? The Poland, that one?
Sure. So let's see.
Professor, I actually wanted to share my screen, just to know if I'm on the right track on exercise on, part 2.
Sure.
Okay, let me see if I can share my screen. Can you see my screen, Professor?
Yep.
Okay, so I was looking at the first one, which is Poland, and then I was taking into consideration this pink one over here.
Mhm.
Yeah, so I counted 4 units in the ocular.
Mhm.
So
Mhm.
then what I would do is the size for size under 10x will be 4 times 12.5, which is 50, right?
Yes,
That will be 50 micrometers.
that's exactly right.
Okay, so I think I'm going on the right track. Thank you,
Yes,
Professor.
and then you want to measure the same one under 40 and 100x too then, right?
Exactly.
Yeah, okay.
Thank you, Professor.
Yep. Do you still want me to go over it or are you good?
Yeah, I'm good. My
Okay,
problem just
Rachel.
solved. Thank
Okay,
you.
all right, awesome.
I have a problem while measuring the pollen. Like when I get to the highest magnification, 100x,
You
the
don't see the
ruler,
whole thing.
yeah, and then the ruler was like too small to see the whole thing. So I stopped at 40x. Is that okay?
Yeah, so you can say that. You can write that in the table itself that it was too large to measure.
All right, thank you. Oh, and then another issue I had was with the green algae. As I zoomed in, it was harder and harder to make sure that I was measuring the same.
Yeah, so the other thing you
Yeah.
can do is you can maybe measure half and then see if you can multiply that. If you don't think you can do it, because you don't know if you're seeing half or not, you can always
Mhm.
say, that you can't measure it. But it shouldn't matter, should it? Right?
No,
The actual
yeah,
size is the same, right? I mean,
right.
that's the purpose of doing this particular activity, that you realize that no matter what magnification you're measuring it under, it should remain the same. It should be approximately the same as long as you're careful measuring it.
Mhm.
And if your original calibrations are accurate.
Okay, thank you.
Mhm.
Professor, do you mind if we go over the ocular division in micrometers just to make sure that I have the right values?
I mean, so it depends on what you have, right? So I noticed for Justin, I believe he had seen 4 units, so it might be that there is some differences between individuals. So you should go with what we saw, and it doesn't have to be necessarily identical, although I am suspecting it to be. Let's
I meant for the first table for the last column. I just want to make sure that I did the calculations right for 40 and 100 objective lens.
How many divisions did we have in the second at the 40x? I don't remember.
We had for the ocular ruler divisions, it was 32 and then 10 for the stage. And I calculated to be 3.125. I just want to make sure that that's on the right track.
Should be, so.
And for the last column, it was 32 for the ocular and then 5 for the stage and 1.5625 for the last
Yeah.
column.
Yep.
Okay.
Professor.
Yes, Rachel.
Sorry, I know I got a lot of questions.
That's
But
okay.
for the, it's also for Poland, the 100x, the ruler is smaller than the Poland. So how do I
Yeah,
calculate?
yeah. So you can either approximate it, but when you did it for 10x and 40x, what is the measurements you got? Are they the same, different, big, small? What is it?
It's the same, also 50 micrometer,
Yeah, so what do you expect under 100x?
also 50.
It will be exactly, but you can't measure it, so you're just gonna say that it was too large to measure.
Oh, okay. Thank you.
Yep.
Professor?
Yes, Vanessa.
For the diatome, does it matter which one we're measuring?
You can measure any one of them, just keep on measuring the same one for the other magnifications.
Okay, got it. Should we specify which one we're measuring, like the longer one or the circular one?
Yeah, you can. Yeah, sure.
Okay, thank you.
Buzzer.
Yes.
So, sorry if I didn't hear what you said, but for the green algae, since it's too long to measure, did you tell us to approximate it if we could?
You can approximate it, or if were you able to at least measure it under 10x and 40x?
It's too long to measure. I mean, for that size, for even a 10x station.
Algae.
Yes.
I just measured a cell. Just
Cell,
one of the
yes,
cells.
one
Yeah.
of the cells on the algae. Don't measure everything, just one cell.
All right.
And I want to follow up about a question on the algae. Like in the explanation, I wrote that my differences in size measurements for the green algae were very different because like I had measurement errors due to low resolution.
Yeah.
Yeah, all right. Thank you. Hi, Dr. Chima. So I am on the paramecium questions.
Yeah.
And then like, do I use the same metric units as the ones from our calibration lab?
That's right.
All right, thank you.
Professor, what do we do once we're done with that?
If you're done with your exercises, we're done. That's it. We had scheduled, but just make sure to pour your plates.
Are we gonna go over the test your knowledge one, or is that for us to do on our
You
own?
should do it on your own.
Uh-huh, okay. All right, thank you.
Yep. Yes, if you're done with the activities, you may leave, guys.
Hi, Dr. Chima. Can I share my screen with you? I have a
Yes.
confusion about a question.
Yeah, I think I know which question it is. I was just looking at it and I think it's not worded correctly, but yeah, go ahead.
Yeah, so it's like determine the length of the micrometer in metric units using the calibration data. So from the top here, I measured 7.5 and it's like 10 ocular units. And from our previous lab, I determined that in under 100 times magnification, each division unit is equal to 1.25 micrometers per division. So that's how I got 93 micrometers as the full length of the paramecium. So that's,
But this is a 10x objective you're looking at.
yeah, but 10x ocular lens with, I also took the 10.
No, but that's, I know, but that's your total magnification. That is not the calibration that you should be like equivalent
Oh,
that you should be using, right? I mean, we're going by the objective lens.
I get it. So in the lab, it was 12.5. So this should be
Yep.
90. Then in terms of B, what
I think
were you looking for?
I think it should say microbe, not micrometer. So that's what the error is. That's what I want to correct. So it shouldn't say determine the length of the micrometer. It should say determine the length of the microbe, which is the paramecium.
Okay.
Yeah, I was just looking at it and I was like, what is that? What am I asking?
So it is also 75 ocular division units if that's like.
Right. So if you're measuring 75, then you are that's the ocular divisions, and then you want to, of course, do it for the 10x first, and then you want to do it for 100x. So I shouldn't say magnification, I should say objective lens. So that's the other error that I need to correct. This is what happens when you're trying to put together something so quickly.
Okay.
I'm gonna fix that. I'm gonna fix that. So it should fix on your end, but you can fix it yourself too.
All right. Thank you.
Thank you. Walter, you're there?
Did you ask for me?
Yes.
Oh,
So
yeah.
I was looking at your assignments and I feel like you're doing something wrong. So I just wanna, you don't mind talking about it now, right?
No, I don't-
Okay. All right. So you're not doing your conversions correctly, or you're not, I'm not understanding. So were you filling out the bottom table where you have the measure the size of like pollen and stuff? So you measured it under 10x and then 40x and then 100x. So I understand that at 100x is too large to measure, but under 10x, how many ocular units big was the pollen? Was it
Let me see. So the pollen was one, well, it was four like
Okay.
ocular blue divisions.
okay? And then you multiplied that by
12.5.
excellent. And then when you measured it under 40x, how many divisions was the pollen?
Like 5, I believe, but let me double check. Oh,
No,
no.
40.
Oh yeah, 40. Yeah. Hold on a second. I mean 20. So it was 20 ocular ruler divisions.
And then you multiplied it by what?
By 3.125.
It shouldn't be that big. How is it possible?
Oh, maybe I miscalculated something. I don't know why I put
I feel like you might have multiplied it by 12.5.
maybe
So
my
you have to multiply it by the correct conversion.
I'm sorry about that. I didn't even
Yeah, no,
realize.
I just wanted you to go back and I just don't want you to repeat that mistake, ever again. So I'd rather that you fix it now.
Okay, yeah, I don't know why I put that number down. I'm sorry.
That's all right. And
Okay,
so what do you have now?
62.5.
Yeah, it's that's better. Are you measuring the same pollen though? You should be measuring the same pollen, so it should be approximately in the same area in terms of its size.
Yeah, it's the same one.
Okay, all right.
Thank you.
Yep.
I have one last clarification before I leave this session.
Yes,
So yeah, for the preparation of the Petri dish, for after lab today, we're just working on what the video instructs us to do. We're not doing the whole instructions.
No,
right?
you're just gonna pour the plates. That's it.
That's it. All right. Thank you. And then, so I've submitted my calibration assignment and the paramecium assignment. That's all there is for today.
Yep.
All right. Thank you. Have a good rest of your night.
Bye-bye.
Bye-bye.